In the name of Allah, the most beneficent, the most merciful. It is me, Saira Arshad from BS Chemistry 1A. This presentation is presented to Dr. Shehbaz Ali. Today, our topic of discussion is genetic code. As we all know, a gene is a specific segment of DNA that encodes the synthesis of particular polypeptide or protein. Therefore, the order of amino acid is in a polypeptide in according to the sequence of nucleotides in any part of DNA. The relationship between nucleotide sequence of amino acid sequence is called genetic code. In other words, the information in DNA for the synthesis of protein is called genetic code, which is transcribed copied into messenger RNA. In simple words, relationship between nucleotide sequence and amino acid sequence is called genetic code. The messenger RNA consists of random sequence of only 4 kinds of nitrogenous bases: adenine, guanine, cytosine, and uracil. Now, here the question arises, how can a codon with 4-letter alphabet specify a protein which at any given point contains one of the 20 different amino acids? Clearly, a single base cannot specify a single amino acid. If one base specifies a single amino acid, then there will be only 4 different amino acids could be encoded for. And proteins containing only 4 different amino acids would be formed. Nor it is feasible for just 2 bases to specify a single amino acid, since only 16 amino acids could be encoded, but 3 bases are sufficient. With 3 bases, a total combinations of 64 are possible. Each is specific for a particular amino acid. Therefore, a triplet of bases along the length of messenger RNA that specify a particular amino acid is called as codon. There are total 64 codons. 3 of these codons act as stop codon. One of must be present at the end of messenger RNA that indicates that the message is over. Since these codons do not encode many amino acids, hence called sense codon. One of these sense codon, methionine AUG, also acts as start codon, which must be present at the beginning of all the sequences that encode for amino chains. Here is the table for genetic code. Genetic codes. Now we will discuss here the characteristics of genetic codes. Some amino acids are only encoded by single codon, while others are encoded by more than 4 codons, and amino acids leucine and serine are encoded by 6 codons. This property is known as code degeneracy. The genetic code is universal. It is the same in almost all the organisms. For example, AGA specify arginine in bacteria, in humans, and all other organisms whose genetic code has been studied. So, genetic code is universal. There is no punctuation between each codon. It means that there is no gap between 2 codons. We can't use a comma, inverted commas, anything else between the codon. The genetic code is non-overlapping. Messenger RNA sequences is not read as separately. It was read combinedly. Thank you.